much of the parietal and all of the occipital lobe, so that you can see some of the structures that are deep to those lobes. And primarily I wanted to show the hippocampus, which is illustrated here, kind of peeled away, and also its relationship to the lateral ventricle and to the basal nuclei and to the thalamus underneath it. And so here I've cut into the telencephalon, the cerebral hemisphere, down to the lateral ventricle. And so what we see here, this space that's right here, is the lateral ventricle. And you can see the choroid plexus that sits in the lateral ventricle here. And you can see how it follows the lateral ventricle around. And it would follow most of the lateral ventricle. And then remember, the lateral ventricles have this kind of C-shaped structure as the hemisphere grows toward caudally and then inferiorly. And the hippocampus and the basal nuclei and the ventricle all reflect that C shape, which can be confusing when you're looking at the coronal sections, because in some of them you'll see some structures both above and below the lateral ventricle, depending on where on that C shape they are. And the hippocampus is one of those that's kind of problematic. Here you can see the cranial end of it, or rostral end of it. And then here it's cutting through the part that loops around. And so you see the hippocampus is this big elongated structure. And if you move back forward again, you can see the hippocampus ventrally. Actually, this is probably the amygdala, but you can see the tail of the hippocampus depending upon where that structure is. So this is the hippocampus here, and the hippocampus is modified cerebral cortex. And so it's hard to tell in these, but if you look at these views, you can see that it's really a continuation of the neocortex, and it's laminated. It has layers of cells and fibers like the cortex, but it's highly specialized. And so on the surface of the hippocampus, here we can see this white layer, which is a layer of axons. And these axons over the body of the hippocampus are called the alveus. And I don't know if they have that labeled in here, but that's this white layer that you see right along the surface. And that is the axons that are coming into and going out of the hippocampus, and that collects cranially or rostrally to form the fimbria. And so this band at the rostral end of the hippocampus, or edge of it, is the fimbria. And they do have the fimbria labeled in several locations on these slides. It's this big band of white matter here. And the fimbria comes together to form the fornix. So the fimbria comes together at an angle and then forms the fornix, and then those fibers will proceed down into the hypothalamus and into the septal region. And the ones that go into the hypothalamus are called the postcommissural fibers of the fornix. And the fibers that go into the septal region are called the precommissural fibers, and that's because they're divided by the anterior commissure, which is this little bundle right here that's coming out at us.
And commissure just means bundle?
Commissure means it goes across from left to right.
Oh, yes, okay.
And so commissural fibers are fibers that decussate or cross from left to right. Mm-hmm. So we have the anterior commissure just rostral to the thalamus, and you can't really see it here, but there's a bundle of fibers right here, the posterior commissure, that's just caudal to the thalamus. And then we have the corpus callosum, which I've torn away up here, but you can see in your sagittal sections, which are a lot of the fibers from the internal capsule that are going from left to right. So anyways, on the surface, we have these layers of fibers. And if we pull up the hippocampus carefully, it forms this C shape that goes down into the temporal lobe, and at the end of it is the amygdala. And so this swelling right here is probably the amygdaloid body right here. And it's very similar to the hippocampus and is linked to it in a number of different functions or pathways, circuits. So we often talk about the amygdala and the hippocampus together when we talk about the limbic system, when we talk about learning and memory, etc.
So I thought the amygdala was like its own little bean-shaped thing,
It
like a little
is.
lima bean. So that's not it, though.
That's it.
That is it.
That's a little bean-shaped thing, swelling right here.
Okay. Little lima bean. Okay.
And if we look at the ventral surface of the hippocampus, a couple of structures beside the fimbria, a couple of structures that are pointed out in your lab book are the dentate gyrus, which in this view is just kind of the tissue in the middle, just caudal to the fimbria. And that's a gyrus just like in other parts of the cortex. And then right next to that, where we have this blood vessel running, you can see that dark streak, that little line, that's the hippocampal fissure. And the hippocampal fissure separates the dentate gyrus from a portion of the hippocampus or structure right next to the hippocampus proper called the subiculum. And you don't have subiculum labeled on any of these,
I'm sorry.
but you can see this tissue right here is subiculum, and then that is attached to the entorhinal cortex.
Do we need to know?
Sorry, what was subiculum?
Subiculum is the side of the hippocampus, tissue right next to the hippocampus. And it's one of the important areas. It's a continuation of the hippocampus, but it's not enclosed within the original hippocampus as it's been described. So one of the things, and we'll see in the lecture text, you'll see the organization of these. Actually, I think they've got a drawing in the back here. If you cut in cross-section the hippocampus, here's the fimbria, the fibers at the cranial edge. Here is the dentate gyrus, and here's the hippocampal fissure. And the subiculum is this area right next to it. So the hippocampus is organized like the rest of the neocortex in that the deep layer is fibers like the internal capsule. And that's the alveus and the fimbria. Those are the fibers that go into and out of the hippocampus. And then this would be the superficial or surface layer of the cortex. And so in one of the deep laminae or layers are pyramidal cells, just like we've talked about with the Betz and non-Betz motor cortex. Those are cells that project out of the hippocampus. Their axons will leave the hippocampus and go into the subiculum or go into the fornix, and that's where the output from the hippocampus goes. Most of the input comes from the subiculum in a set of fibers called the perforant pathway that goes to innervate these interneurons, which are granule cells and mossy cells, just like in the parts of the cortex that we've talked about already, like the visual cortex, where you have incoming input from the lateral geniculate nucleus, etc., and the interlayer connections.
How do you spell subiculum?
Subiculum.
Gotcha. Okay.
I have to get to micro.
Okay.
Boy.
So anyways, I wanted to show this because later, and we will spend a little time talking about learning and memory and the hippocampus, and we'll talk about these pathways. This is where long-term potentiation was discovered and has been studied intensely. There's a lot of glutamatergic synapses here, and so these cells are the like textbook cells that respond to repeated stimulation with hypersensitization or facilitation.
So if you're like long-term prone to stress or something, that's Is that what you're talking about, or?
No, I'm talking about the actual changes at synapses that occurs when you learn something. When the
Oh.
synapses are strengthened, that's long-term potentiation.
I see.
And that's it was discovered involving these synapses, and it probably occurs throughout the whole cortex.
It was like long-term
Wherever there's
memory.
glutamatergic synapses. Now, the other thing I wanted to show you guys on this are some of the thalamic structures underneath that. And so, if we move the hippocampus out of the way, we can see, I want to do this without tearing it, we can see the midbrain, superior colliculus and inferior colliculus right here. And we can see the thalamus, at least the dorsal and lateral thalamus, up underneath the cortex. And there are 3 prominent bulges in this part of the thalamus. One that's sticking out towards the hippocampus, kind of caudally, is the pulvinar. And you'll see the pulvinar on your coronal sections at the very caudal edge of the thalamus. And then as we move more laterally, we can see the medial geniculate nucleus and the lateral geniculate nucleus. And one of the keys with the lateral geniculate nucleus, if you follow the optic tracts from the chiasma around the brainstem, the optic tracts come up and much of the fibers terminate here in the lateral geniculate nucleus. And so that's the swelling that's up here right at the end of the tract. And then a lot of the fibers that don't synapse there will continue into the midbrain and go to the superior colliculus. And so these 2 structures here are the primary targets of the ganglion cells from the retina. And then the other thing I wanted to show you guys on this, just for perspective or context, is that when we look into, so the hippocampus is on the kind of the caudal end of the lateral ventricle. And if we look at the lateral or ventral surface of the lateral ventricle, we see this big swelling here. That's the caudate nucleus. And so the basal nuclei that we'll start talking about tomorrow are located right under here. This is the surface of the lateral ventricle. And beneath that, this big swelling is the head of the caudate nucleus. And as you move more caudally, that narrows down to form the tail of the caudate. And so when you're looking in the coronal sections,
And that makes like a C with the hippocampus, but like
Yes.
the other way.
so if you're looking at the coronal sections, let's move the next one forward here. Oh wait, forward is the other way. You can see the caudate nucleus start out in the forebrain pretty large as the head of the caudate, and it's right next to just lateral to the lateral ventricle. And then as you move caudally, it narrows down to form the tail. And the tail also is proceeding caudally at the edge of the lateral ventricle. And this is about the same place that the hippocampus is becoming visible. And so if you think about this view, the caudate narrows down out here lateral to the ventricle, and the fornix is coming up and enlarging as the hippocampus as you continue to move caudally, and then the hippocampus wraps around. So underneath the caudate nucleus are the globus pallidus and the putamen, the rest of the corpus striatum. And so if we look at these guys in here, these are the basal nuclei right here. Globus pallidus, putamen, and caudate. And this is the corpus striatum. When we talk about the dopamine fibers from the substantia nigra, that nigrostriatal pathway, the dopamine's being released in here. And that's what we'll be talking about for the next few days. And that's this bulge underneath here. Got it?
Mhm.
So this helps. I think it helps when you're going through these to be able to envision how one structure is decreasing in size or another one's increasing in size. And it's also real important when you get back to the midbrain that you can think about the lateral ventricle curving around in that C-shaped structure and then progressing forward down in the temporal lobe.
So, when we relate a sheep brain to a human brain, how, if you like took a sheep brain and like bent its spinal cord like a 90-degree angle?
So we have a flexure, we have a mesencephalic flexure, so that our brain axis curves
Yeah,
almost 90
almost
degrees.
like that.
So the spinal cord, medulla, and pons,
Right.
and up to about the midbrain are oriented along our longitudinal axis, and then the diencephalon and telencephalon are curved forward.
Okay. So if you took a sheep brain and just bent its brainstem, it's essentially what our brain would look like. Okay.
Yeah.
Okay.
The difference is, so a lot of these structures and their relation are very similar. The difference is the relative sizes of these structures. So
Sure.
especially in the cerebral hemispheres, the different lobes are very different,
Size-wise.
yeah, correlated to the type of thinking that sheep do and humans do.